Hello everyone. First of all, I would like to thank the organizers for inviting me to this wonderful meeting. Today, I will present our recent work about the neuronal basis of healthy aging. As you know, aging is the main risk factor for many chronic diseases such as diabetes, heart failure, Alzheimer's disease, and so on. So understanding the biology of aging is very important for preventing this kind of aging-related human diseases. In our study, we're using elegans as an animal model because elegans has a clear genetic background and is easy to genetically manipulate. Studies using elegans have won 4 Nobel Prizes, and elegans is especially classic for aging research because elegans has a short lifespan and many important longevity pathways has been firstly identified in elegans and then found conserved in mammals, even in human beings. Our research interest mainly focuses on the mechanism underlying the regulation of aging-related behavioral decline. So there is a lot of different types of animal behaviors which is regulated by different types of neurotransmitters. To narrow down our study, we just focus on those animal behaviors regulated by serotonin and dopamine. We firstly examined the 2 important neurotransmitters, serotonin and dopamine level in aged elegans, and we found the level of these 2 neurotransmitters significant decline during aging, as evidenced by immunostaining and HPLC results. We also found that the expression level of the shared synthetic enzyme for serotonin and dopamine, called BAZ1 level, declined during aging in elegans, and this decline is responsible for aging-related loss of serotonin and dopamine level in aging elegans. Aging-related loss of serotonin and dopamine causes decline of many important animal behaviors, including male mating behavior. Male mating behavior in elegans usually regulated by serotonin and dopamine. During aging, the male mating behavior significantly declined, and we found that if we added serotonin in cultivating plates, aged male show significantly enhanced male mating ability, suggesting that the serotonin and dopamine play a very important role in aging-related behavioral decline. We then propose if a gene or a drug can abrogate the function of neurotransmitters in aging animals, then this gene or drug may can prevent aging-related behavioral decline, or may can prevent neurodegenerative diseases. We then performed higher throughput drug screening, and we had screened about 10,000 compounds. Finally, got 11 compounds that can abrogate serotonin and dopamine level in aging elegans. And we further found that these 11 compounds indeed can improve animal behaviors in aged elegans, including eating behavior and locomotion, suggesting that this drug may can prevent behavioral aging. We also performed a genome-wide RNA screening for clones that can increase serotonin and dopamine function in aging worms, using BAZ1 expression level as a genetically traceable aging marker. And we selected these clones that can enhance BAZ1 expression specifically in aged worm. So we had screened about 16,000 clones, and finally we got about 60 candidate genes. Among them, 10 of them has been reported to be related with cellular senescence and neurodegenerative diseases, and there remain more than 40 genes. Then we want to know whether this newly identified gene indeed regulates behavioral aging or not. We focus our study on two epigenetic regulators, BAZ2 and SET-6, for further study. We then generated BAZ2 and SET-6 knockout worm and performed a behavioral test. We found that deletion of SET-6 or BAZ2 or both can significantly enhance the pumping rate in aging cell organ, suggesting that deletion of these two genes improve eating behavior in aging worms. We also found that deletion of BAZ2 or SET-6 or both significantly enhanced male mating behavior in aging males. And we further found that BAZ2 SET-6 knockout worms showed significantly extended lifespan, suggesting that BAZ2 and SET-6 indeed regulate behavioral aging and lifespan in cell organ. We then ask what's the mechanism. So the domain of SET-6 is predicted to have H3K9 methyltransferase activity. Then we express and purified the cell domain of SET-6, then performed enzyme activity test, and we found that the cell domain can enhance H3K9 ME2 and ME3 level. And we also found that in BAZ2 SET-6 or double knockout worms, the H3K9 ME3 level significantly decreased. Together, all the data suggested that BAZ2 and SET-6 affect H3K9 methylation level in cell organ. We then performed RNA-seq to adjust how BAZ2 and SET-6 affect gene expression. Interestingly, we found most of upregulated genes in SET-6 and BAZ2 are overlapped, while only few downregulated genes in SET-6 and BAZ2 are overlapped, suggesting that BAZ2 and SET-6 work together to suppress gene expression. We also performed ChIP-seq analysis, and we found that BAZ2 occupied DNA region and the SET-6 occupied DNA regions are mostly overlapped. Furthermore, we found overlapping upregulated gene and BAZ2 SET-6 co-occupied DNA regions are mostly enriched in the promoter of genes related mitochondrial function. Then we ask whether BAZ2 and SET-6 affect mitochondrial function. Indeed, we found that deletion of BAZ2 or SET-6 significantly enhanced ATP levels and oxygen consumption rate, while overexpression BAZ2 or SET-6 significantly reduced ATP levels and oxygen consumption rate, suggesting that BAZ2 and SET-6 repress mitochondrial function. Mitochondrial unfolded protein response, called mitochondrial UPR, plays a very important role in mitochondrial homeostasis and longevity regulation. Then we ask whether BAZ2 and SET-6 modulate behavioral aging through mitochondrial UPR. Indeed, we found that deletion of BAZ2 or SET-6 or both significantly enhance the activation of mitochondrial UPR. This activation was abolished by UBL5 ATFS1 RNAi. The two gene is a positive regulator of mitochondrial activation. Interestingly, the improved behavioral performance in SET-6 BAZ2 or double mutant worms were also abolished by UBL5 and ATFS1 RNA, suggesting that UPR, mitochondrial UPR is required for behavioral preservation in aging animals. Next, we want to know whether the BAZ2 SET-6 modulate behavioral aging is conserved in mammals or not. BAZ2 has two mammalian homologs, BAZ2A and BAZ2B. The set domain of SET-6 is similar with the set domain of EHMT1 and EHMT2. We then analyzing the published human brain gene expression data set, and we found the expression level of BAZ2B and EHMT1 significantly enhanced during aging, while the expression of BAZ2A and EHMT2 did not change during aging in human brain. Then we focus our study mainly on BAZ2B and EHMT1 in mammals. Interestingly, we found that the expression level of BAZ2B and EHMT1 is upregulated in brains with Alzheimer's disease, and their expression level positively correlates with AD progress. Furthermore, we found that their expression level negatively correlates with the expression level of some important mitochondrial proteins, suggesting that BAZ2 and EHMT1 may also can regulate mitochondrial function in mammals. Indeed, we found that knockdown of BAZ2B and EHMT1 improve mitochondrial function of cultured neuron, as evidenced by enhanced ATP level and oxygen consumption rate in cultured neuron. Furthermore, we found that overexpression of BAZ2B and EHMT1 reduced mitochondrial function, as evidenced by reduced oxygen consumption rate and ATP levels in cultured neuron. We then generated BAZ2B knockout mice. Consistently, we found that the mitochondrial function in BAZ2B knockout mouse showed significantly enhanced mitochondrial function in hippocampus, cerebellum, and some other brain regions, suggesting that BAZ2B suppress mitochondrial function in vivo. We then examined the aging-related phenotype of BAZ2B knockout mice. Interestingly, we found that all three genotype mice show similar body weight at the young adult stage. As you know, when mice getting old, they will gain some body weight. And we found that aged BAZ2B knockout mice show significantly low body weight than age-matched wild-type mice, suggesting that BAZ2B regulates aging-related body weight gain, and also BAZ2B regulates body energy metabolism. We then performed a novel location recognition test. In this test, we put a mouse in a box with two objects, and the mouse show similar interest with the two objects, and they spend equal time with the two objects. Then we replace one of the objects, and the mouse will show more interest with the replaced object, and they stay more time with the replaced object. And we found in this test, all three genotypes of young adult mice show similar ability to recognize novel location of the objects. And in aged wild-type mice, they lost this ability to recognize novel location of objects, while BAZ2B knockout mice have maintain very clear ability to recognize the nerve location of object, suggesting that abolition of Baz2b preserves cognitive function in aged mice. So finally, we're giving a working model here. During aging, the expression level of two important epigenetic regulators, BAZ2 and SET-6 expression level significantly increased, then resulted in enhancement of H3K9-ME3 level, then repress the expression of genes related to mitochondrial function, then reduced mitochondrial function, caused aging-related functional decline. When we downregulate BAZ2 or SET-6, then can reduce H3K9 ME2, ME3 level, then can upregulate gene expression related to mitochondrial function, enhanced mitochondrial function including mitochondrial UPR activation, then can delay aging. Summarized here, in this study, we provide first global view of genes that may regulate behavioral aging, and we found that BAZ2 and BAZ2B and SET-6 EHMT1 prevent healthy aging, and these two epigenetic regulate mitochondrial function and cognitive performance through affecting H3K9 methylation level. And we hope that BAZ2B and EHMT1 can be a novel anti-aging gene that will help us to develop approaches to anti-aging-related behavioral decline. Finally, I want to thank all people in my lab, especially these graduates contribute to this project. And I also want to thank the collaborators, and also thank to funding resources. I want to stop here, and I'm ready for questions.